My name is Margaret DeAngelis, and I am a professor at the University of Utah, the Moran Eye Center in the Department of Ophthalmology and Vision Sciences. I am also adjunct faculty professor in the Department of Pharmacotherapy in the Department of Health, Population Health Sciences. Thank you for having me here today. I'd like to talk about some of the research that's going on in our lab. We've just had a paper published recently on our standardizing phenotyping for a donor eye bank. And by the term standardizing, I don't only mean the rigorous phenotyping and characterization of the eyes, but also quality control on the accessioning and processing of the tissue. This is a wonderful gift that organ donors give us, and it's very important to have the tissues handled and processed within 6 hours of postmortem. This is because as the eye is left out in the open outside of the body, it begins to degrade and it releases stress-related genes. And those likely are not associated with the pathophysiology of age-related macular degeneration or glaucoma, but it's due to tissue changes that happen upon exposure to air. So as a result, we want to start with good quality control so that we have robust downstream experiments and that we know that the results we're getting are truly due to the pathology of the disease and not to just random environmental factors.
How are these eye tissues used?
So one of the things we're trying to do, I'm doing with my collaborator, Michael Farkas, is to make induced pluripotent stem cell lines as well as primary cell lines from the tissues of the donor eye. And the reason we're doing this, as you know, animal models have not been ideal either for age-related macular degeneration or glaucoma, two of the leading causes of blindness in the aging population. What we want to do, and we indicated this in our manuscript, Dr. Leah Owen is the first author on that, we indicated that we want to be able to make these in vitro assays, eye in a dish, and to be able to compare it back to the actual tissue in the back of the eye from the same donor. That way we're actually knowing, we directly know that we are replicating what is going on in the human body. Because the further you get away from the human body, you could lose attributes of what's really going on in the local environment, meaning the eye. So we'll also be making cell lines, the induced pluripotent stem cells, from blood. And the reason why we can't use direct eye tissue is you need these assays to check and do experiments to test disease mechanism. And if you can get to disease mechanism, you can get to therapeutic targets. So we're doing things right now with our eye tissue like epigenetics, looking at environmental factors, methylation, chromatin, within the chromatin changes within our genome. We're also looking at gene expression, what's being regulated, what goes up, what goes down between disease and non-disease states. And more importantly, we're looking at different disease stages, early to late stage. And because we have a wonderful group of clinicians, scientists we work with, namely Leah Owen, Dr. Ivana Kim from the Mass Eye and Ear, Al Vitelli, and Dr. Akbar Shakur from the Moran Eye Center on the Retina Service, we're able to make sure that these eyes are rigorously phenotyped so that it replicates what's going on in the clinic, what the actual physician or ophthalmologist sees in the clinic, so that our work can have more impact translationally. Well, the cell lines that are out, there are no adult RPE cell lines that are out that are commercially available. You can call it an adult cell line as long as it's not embryonic, but it could really be a fetal cell line. And to me, that's disturbing because a fetal cell line isn't ideal for studying an age-related disease on. So one of the reasons we're doing what we're doing is we want to make primary cell lines as well from our eye tissue so we have adult cell lines. We want to compare those adult cell lines from the retina pigment epithelium and the retina to the induced pluripotent stem cell lines and then the actual original tissue and the blood, all from the same donor. We also collect protein on the patient in the form of serum and plasma. And this is the central dogma of genetics is you go from DNA to RNA to protein. And at any one of those points in that pathway, something could be going wrong. So we think by covering all our bases, we can get to a therapeutic target, but we really need a model in a dish. And many people, including our group, have developed organoids within a dish. However, that more faithfully replicates the fetal retina. And that's great for studying inherited retinal degenerations, including retinitis pigmentosa, Stargardt's, and Best disease. However, we really need to make an in vitro assay that's recapitulating the adult disease. And I think this is the good way to go.
What are your next steps in creating a viable model?
Right now, along with my collaborator, Dr. Michael Farkas, we're making cell lines. So we're making primary cultures from the retinal pigment epithelium and the retina from both diseased and non-diseased patients. We're also taking the bloods from these same patients, these same donor patients, and making induced pluripotent stem cell lines. And then with some of our findings, we've developed from using a systems biology approach to all our analysis, and that is taking the gene expression changes, so the activity, measuring the activity of a given gene, along with the DNA part and the epigenetic part, and the DNA part, whole exome, whole genome sequencing on these eyes, and putting that all together to tell a story. Because if you have evidence for more than one line or one pathway, that helps you to really target a therapeutic target. And it's also a way of validating and replicating your data, which is extremely important if any of these therapeutics are going to have global applicability. Along with that, so we're taking our findings from that, and we're knocking in or knocking out the alleles or the variants that we think are associated with the disease risk or progression of disease, and seeing what this does to the actual eye in the form of a cell line, because you can't do this in just a standard donor eye. You have to make it available in an assay that you can actually manipulate rigorously and scientifically to get to your answer. So that is what we're doing.
How do you think that this research will influence patient care?
What we hope is by having all this evidence pointing to one direction or pathway, we can identify a therapeutic target. We can then go to and hopefully say, this is our data, and they can do testing. Then we would go to a GFP site and have them test it to see what compounds maybe target this gene or protein, and what else you can do toxicity tests before you even get into testing it in an actual human or an animal model.
Do you have a message for anyone who may be considering donating their eyes?
Yes, please contact me directly, and I will put you in touch with our eye bank. To ensure the freshness right now of our eyes, we only collect donor eyes that are within an hour driving distance of our research lab. Unfortunately, we do not take eyes from the outside unless they're fixed or processed in a certain way, and they have to be within 6 hours. Unfortunately, many things published in the literature and that are available out there do not tell you the post-mortem time of processing. They also don't tell you the phenotype. Does the eye have glaucoma? Does it have age-related macular degeneration? Does it have diabetic retinopathy? And all these things need to be taken into consideration, because somebody with diabetes is going to be at risk for diabetic retinopathy. That is a donor eye that's important for another study, but we could not use that eye for a normal control in a study like age-related macular degeneration, because you could see genes that are involved with diabetic retinopathy that have nothing to do with the pathophysiology of age-related macular degeneration. So you have to have very rigorous control and know where your eyes are coming from. Another thing we find is that while we're able to match the diagnosis in most cases, in 100% of cases, with what has been previously diagnosed, there's a lot of patients walking around in the general public who do not know they have age-related macular degeneration and have the early forms. And we do not see that until we get their donor eye. Particularly from areas where you have one main eye center and not everybody may have seen an eye doctor in their lifetime. And that's a way we can diagnose, and we see there's a lot more people walking around with early age-related macular degeneration, and those are very important to our study because we'd like to identify a therapeutic before the patient progresses and has already lost vision. Those are the people we want to reach. We don't want to ignore the people that are like we're studying them, but we want to see if we can stop or prevent the disease before it goes to end stage.